Okay, welcome everyone. You're joining the three National Service Centers sponsored by the NIH Common Fund's Transformative Cryo-EM program. This webinar series is called the Current Practices Webinar Series, where we show you the best tips and tricks and also highlight the users that have made use of the National Center program. This is a regular webinar on the last Thursday of every month. The next one will be Thursday, June 29th. More details will come soon. The speaker today is Akif Tezcan, that's invited by S2C2. My name is Ed Ng. I'm the manager of NCCAT, and I'm joined by my colleague, Mike Schmidt from the Stanford SLAC Cryo-EM Center. Shortly after a few housekeeping updates, we'll turn it over to the three National Service Centers to give an update on the resources offered to you at no cost across the nation by the NIH Common Fund. Today's webinar will be recorded. It'll go on to our common website at cryoemcenters.org, where you'll find more information on the events coming up. So I'll turn it over to our colleagues to introduce the relative national centers. I don't quite see my colleague from PNCC, the Pacific Northwest Cryo-EM Center, but they offer no-cost services and training, and they have a very robust, not only workshop program, but embedded training program. They have 4 dedicated Krios instruments for instrumentation access and screening capabilities with recurring access on their Arctica. To find out more, please visit their website. I'll put on my other hat to highlight NCCAT, that is the National Center here in New York. We also offer a robust cryo-EM access, instrumentation, screening and training program. Something that I would like to highlight, as you notice, this is May, a lot of people are graduating. We've had a lot of trainees graduate. And something to make note of is that the 3 national centers is part of a network of resources across the nation. A lot of our trainees are now in Michigan with Mike Chinfroco on his cryo processing workshop. So if you want to find out more, not only on the national resources at the 3 service centers, but at the full network, you can go to nccat.nysbc.org or our common website. And I'm going to turn it over to Mike to introduce SLAC, as well as introduce our speaker.
Hi, this is Mike Schmidt from S2C2, the Stanford SLAC Cryo-EM Center, where we have 4 Krioses and a Talos Arctica for screening. We, like the other 2 centers, offer training, we offer screening, and we offer high-resolution data collection. We offer workshops. We just finished a workshop last week on image processing through the package called Scipion from Spain, which incorporates all of the different image processing packages into one. And it was a very, very successful hands-on and remote experience for everyone. Join us at S2C2. There's the website, and we're looking forward to seeing you at S2C2. Now I'd like to introduce our speaker, Akif Tezcan from the UCSD Department of Chemistry and Biochemistry. He's a professor of chemistry and biochemistry. He was a recipient of a Burroughs Wellcome and Helen Hay Whitney postdoctoral fellowship at Caltech from 2001 to 2005, as he received his PhD from Caltech. He has his own group now at UCSD that's concentrated on understanding metals, but I believe he'll talk about other things today. And his seminar today is, we see the name of the seminar, excuse me.
Yeah. Here, I'll make it easier for you.
Yeah, that'll be great. Ah, yes, protein assemblies made by evolution, then by design, ellipsis, and understood through Cryo-EM. Thank you, Akif.
I'll pay back the services of S2C2 by making the introduction easier for you.
Okay, that's great. Thank you. It helps us. And we enjoyed your time here too. Okay, thank you.
Should I get started?
Yes, go ahead.
All right, Mike, first of all, thanks for the invitation. And of course, I'd like to thank, the DOE facilities, especially at Stanford S2C2, that made a lot of the research I'm going to talk about possible. Here's a view of San Diego. This is a sunny picture, but we're in the May gray area, time of the year now. So it's all gray, but just to orient you, this is the Pacific Ocean. This is the famous Geisel Library. And I'm pointing at the office where I'm sitting right now, and the oldest building on campus that dates back to 1960. That's called the Urey Hall. In any case, like Mike said, I am not a Cryo-EM specialist or a structural biologist. But as the scope, because of the scope of our research and the directions it's taken over the years, Cryo-EM is becoming a more integral part of it. So what I'm going to try to do is to tell you two stories that are somewhat disparate, and I'll try to force a connection between them, where Cryo-EM has been critical for what we're doing, and like most of you in the audience, and even an inorganic chemist like me, a lot of inspiration comes from living systems, like this bacterial cell that is jam-packed with molecular machines and macromolecules. And when you think about the complexity of cell, you think that most of this complexity, chemical complexity, derives from proteins and protein assemblies. And actually, supramolecular architectures that are composed of multiple protein domains. And those come in two different flavors, as I like to think about. One are the oligomeric structures that are discrete, and these are the machines that biology uses to carry out complex biochemical tasks. And here, as I'm giving as an example, three of the perhaps most important enzymes in the biosphere that are responsible for photosynthetic water oxidation, nitrogen fixation, and also oxygen respiration. These are, of course, because of my bias, also metalloenzymes. And they're composed of multiple domains that, like moving parts of a machine, carry out complex multi-step reactions that you could not do with just a small single domain protein. And the other flavor of protein assemblies are materials or templates that are biologically less complex. They're made out of few building blocks, but through self-assembly in discrete dimensions, they carry out very important functions such as containment, movement, directional movement, protection, or even because of the sizes of these machines and their dynamics, and they're also their dimensionalities, they present hard subjects for crystallography, for example, and also spectroscopy. So this is where Cryo-EM becomes very important. So our overarching goal is to understand the mechanistic details of natural protein-based machines that evolution made, which would be a top-down approach, that typical biochemistry approach, but also to be able to build such protein-based assemblies from scratch, and that is the bottom-up part of my group. And this is more the chemistry approach. Can we actually make something to understand it better? And again, as I said, the technical challenge in these problems is that such assemblies, multi-component assemblies, are highly dynamic, and they have structures and dimensionalities that are not compatible with X-ray crystallographic characterization. And that's why Cryo-EM is so essential. So the biological machine that we work on, we concentrate on, is nitrogenase, that I'm going to spend the first part of my talk on. Nitrogenase is the only enzyme that is capable of taking atmospheric nitrogen and reducing by 8 electrons into ammonia, which is the bioavailable form that can be used for making amino acids and nucleic acids, for example. And this is also a costly process that requires 16 ATP molecules for each turnover cycle. Now, this protein machine is quite large, and it's composed of 2 component proteins. One component is the MoFe protein, that is the catalytic protein, that is an alpha-2 beta-2 heterotetramer that has C2 symmetry. And this is the catalytic protein that fixes nitrogen. And the second component is called the iron protein because it contains an iron sulfur cluster, that is the ATPase. And as shown in this crystal structure that is stabilized by a non-hydrolyzable ATP analog, two iron proteins bind to the symmetrically related surfaces on the C2 symmetric MoFi protein. Now, I'll warn you ahead of time that this is a crystallographic picture, and as I'll show you from the Cryo-EM structures, this is actually not what's happening in solution. And also keep in mind that this is a transient complex between the two proteins, so this has to cycle over and over again. So taking a look at only one of the equivalent halves of this complex, you have now the iron protein on the top and the MoFi protein, one half of it on the bottom. And an iron protein is an ATPase, but also reductase that binds two ATP molecules and has its 4-iron 4-sulfur cluster that docks against the catalytic MoFi protein, which is the catalytic component that takes up those electrons and reduces nitrogen. And iron protein, despite its modest reduction potential, so this is not a very negative potential, it's the only known physiological electron donor that can activate catalysis by the MoFi protein. So there's something very specific about these protein-protein interactions that are important for electron flow across these iron sulfur clusters. Now, as I said, iron protein is called iron protein because it has this canonical ferredoxin 4-iron 4-sulfur cluster that can store one electron at a time, and it injects it upon docking to the MoFi protein into this very unusual P cluster that acts as an intermediary from where the electrons go into so-called FeMoCo, which is an iron, molybdenum, sulfur, and carbide cluster where nitrogen binds, and it gets reduced, and it also has an obligate homocitrate cofactor right there. And of course, you may imagine that the electron flow happens from the iron protein, from the 4-iron 4-sulfur cluster to P cluster and FeMoCo, but this is not the temporal sequence where that it happens. Actually, upon binding of iron protein to MoFi protein, there's a conformational change whose nature is not known, which opens a gate from the P cluster to FeMoCo, where the first electron goes from P cluster to FeMoCo, and that hole in the P cluster is then filled by the electron from the 4-iron 4-sulfur cluster, and that's called the deficit spending model. Okay. So despite essentially 5 decades of research on nitrogen fixation and all the information that we've gathered, the essential questions about nitrogenase, are mechanistic questions, are not really understood in detail. First one is, what is the mechanism of nitrogen reduction? Where does nitrogen bind to this cluster? What are the intermediates of this 8-electron catalytic reaction? Something that we don't know. But the corollary to that question is, why does nitrogen fixation require such complex, biosynthetically expensive metalloclusters, right? There is no analog of these clusters in nature, and it takes a lot of energy to make these clusters. And the second one is, what is the mechanism of ATP-dependent energy transduction in this process? And the corollary to this is, why does nitrogenase require ATP hydrolysis for controlling multi-electron, multi-proton transfer steps when other redox enzymes don't? For example, cytochrome C oxidase or sulfite reductase and nitrite reductase, they also carry out multi-electron, multi-proton transfer steps, 6 electrons and 6 protons, 6 electrons and 8 protons, but none of these require an ATP to do that. So what is so special about ATP-dependent electron transfer? So that's what we'd like to understand. But the problem is, the practical challenge in studying nitrogenase is that the ET electron transfer reactions and ATP hydrolysis are difficult to synchronize. So this is just a cartoon illustration of electron flow in nitrogenase, and we're starting with the iron protein, which is a homodimer and the iron sulfur cluster in the interface. And it takes up an electron from a ferredoxin. It binds 2 ATP molecules, undergoes a conformational change, that activates it for docking against the catalytic protein, upon which it transfers one electron and hydrolyzes these two ATP molecules, and then dissociates. And this cycle has to repeat 8 times, so 16 ATP molecules, whereby the 8 electrons and 8 protons accumulate on the catalytic site with many intermediates in between to reduce nitrogen into ammonia. The problem is, if we don't have ATP, this cycle goes back to the resting state, because when there's no nitrogen, there's no ATP, all of the intermediates release hydrogen from the protons and they go back to the resting state. So unlike normal enzymes that we think about, where you can put an inhibitor or a substrate analog and it sits nicely in the active site pocket that you can crystallographically characterize, this is not possible to do in nitrogenase. So catalytic activity at any given time requires a complete turnover condition. So you need the MoFi protein, you need the iron protein, you need ATP, you need an ATP regeneration system. So this machine has to be working all the time with 2 proteins, which one cannot realize in a crystal lattice. So from crystallographic studies, we cannot glean what is happening at the catalytic site. So if you take those turnover conditions and you look at a solution, you may liken it to a very, very heterogeneous bean soup, right? Where there are many, many different species at any given time in your Eppendorf tube where the reaction is taking place. You can never make it homogeneous as you would do in crystallography experiment, right? That's the challenge. So what is the mechanism? What do we know about the mechanism of nitrogen reduction? Well, we can understand it in the platform in the context of the Thornley low cycle, where the Phi-MoCo in the resting state, which is designated E0, can take up 1 electron at a time and 1 proton to make E1, and next electron gives you E2 with 2 protons, etc. And the cycle takes up 8 electrons and 8 protons. And remember that each of these electron transfer steps also requires hydrolysis of 2 ATP hydrolysis reactions. And what we know is from spectroscopic evidence that the E4 state, which is reduced and protonated 4 times, is critical for the commitment of nitrogen for binding the Phi-MoCo so that it can be reduced. So you need 4 electron and 4 proton transfer steps to even bind nitrogen. And we also know that the so-called belt sulfurs, these 3 sulfurs that are in the middle, can actually be exchanged, come in and out, perhaps to allow substrate binding. But we don't have any crystallographic evidence for what these E1, E2, E3, E4 states are, because as I told you, you need turnover conditions. And anytime you don't have iron protein and MoFi protein, these electronated and protonated species return back to the E0 state by evolving hydrogen. Okay, so how do we capture these states? And the second question, as I told you, was why is ATP hydrolysis required for electron transfer, and what is the mechanism of energy transduction? And we know that this requirement is not purely redox thermodynamics, where you can say ATP maybe decreases the reduction potential of the iron protein and makes it a better donor to reduce Phi-MoCo, because much stronger reductants with potentials that are lower than -1 volt actually don't do what iron protein does. So that means there's something specific about ATP-bound iron protein to activate MoFi protein. So to understand that, when I was a postdoc a long time ago, I wanted to see if I could capture the structure of iron protein bound to MoFi protein in different ATP hydrolysis states. So this is one crystal structure that was obtained, MoFi protein in the presence of iron protein with no nucleotides. And you can see that the iron protein is kind of tilted to the side towards the beta subunit of MoFi protein. This is just 1/2 of the whole complex. And upon binding in a nucleotide, non-hydrolyzable analog of ATP, the iron protein moves to the center. And upon hydrolysis in the ADP-bound state, the iron protein moves to the side. And when we actually look at the locations of the iron sulfur clusters and different nucleotide states, what you see is that as the protein binds ATP, it moves the iron sulfur cluster closer to the surface of the MoFi protein, whereby the distance from the iron sulfur cluster to the intermediary cluster, P cluster, that receives the electrons decreases by 5 angstroms. So that makes electron transfer about 1,000 times faster. So you can say that by modulating through in an ATP dependent fashion, the intercluster distance, you can turn on and off electron transfer. So you can activate catalysis that way. The problem is that this does not explain the mechanism by which a gate is opened between the P cluster and Phi-MoCo upon binding the iron protein, because as we said, that is necessary for electron transfer and catalysis. And when we look at all of these structures and superimpose them, we don't see any changes in the MoFi protein that would imply such a conformational gate. So the lesson that we've learned is that, hey, when you just have static frozen crystal structure, even in different nucleotide states, we may not be able to capture necessarily what is going on in solution during turnover. So that requires us to actually take a look at that bean soup as it's happening, the very, very active turnover condition, and try to see if we can understand the structures of the intermediates that are populated in solution. So that's where we turn into Cryo-EM. And this was a COVID project where one of my student, Hannah Rutledge, who was studying nitrogenase, wanted to learn Cryo-EM and wanted to basically, get the structures of nitrogenase components by Cryo-EM. But we had a discussion saying that what would be the use of doing Cryo-EM of nitrogenase components just to verify the crystal structures. And we decided it's best to see if we can actually do the Cryo-EM of nitrogenase under turnover conditions so that we can get an idea about what the different actives or catalytic intermediates are. So we sought also the help of Mark Herzig and his group, who are Cryo-EM specialists. And Hannah and Mark and Brian Cook and his group were able to devise a nice freezing strategy under anaerobic conditions. So one thing I didn't mention is that nitrogenase is very oxygen sensitive. And we were able to freeze nitrogenase under turnover conditions, under anaerobic conditions, and through a manual plunging apparatus that Mark developed, we were able to freeze the turnover solutions 30 seconds after initiation of turnover, where we reach steady state. So then the problem is, when you have such a soup, how do you actually categorize and characterize the individual species that are populated in the solution? So that took a lot of data collection at S2C2 and a lot of analysis. And since I'm not a Cryo-EM specialist, I'm not going to take you through all of these very arduous steps that Mark and Hannah went through. But suffice it to say that this required about 15,000 movies, about 20 million particles, of which about a quarter were usable. And when we did the analysis, when Mark and Hannah did the analysis of the soup, they saw that about 60% of the particles were just free uncomplex MoFi protein, the catalytic protein. 5% were free iron protein, the electron donor, and 35% were one-to-one iron protein, MoFi protein complexes. So remember the first crystal structure I showed you, where each MoFi protein was bound to 2 iron proteins symmetrically. And what we saw was that is never, ever observed under turnover conditions in solution. So that was already something very, very interesting we got excited about. And it turned out that some of these species could be classified and processed. And as a result, Mark and Hannah and Brian were able to get 3 high resolution structures out of this mixture. And keep in mind that these three species do not necessarily cover all the complexity of the intermediates that are formed in solution. This is just the three that we could in silico purify and get high resolution structures of. And the free MoFi protein, when it was C2 symmetrized, it was 1.8. Without C2 symmetrization, it gave us 1.9 angstrom resolution. And I believe that's probably the highest resolution, one of the highest resolution non-apoferritin cryo-EM structures. And we also were able to determine the structures 2.3 angstrom of two 1-to-1 turnover complex structures, where the iron protein here is in green, and this is the MoFi protein with its C2 symmetry axis that you can see in the hole here. Now, you can think that these two turnover complex structures are about the same, but they're actually different because the iron protein assumes distinct conformational states. So when we look closely at the nucleotide occupancy of these turnover complexes, what we see is that one of them is doubly occupied with magnesium ATP, where you can see all three phosphates, including the gamma phosphate, the structure. And this is the first magnesium ATP bound iron protein MoFi protein structure that we captured. Normally, this is hydrolyzed right away. And then the second turnover complex had one partially hydrolyzed gamma phosphate, and the nucleotide that was across it did not have any gamma phosphate. So that was magnesium ADP. So these structures told us that we're really capturing the nitrogenase during the process of turnover, because these are by default magnesium ATP ADP bound structures cannot be populated right under steady state or under non-turnover conditions. So this also told us that the MoFi protein, although it's C2 symmetric, the two halves of the MoFi protein that bind iron protein are not independent, because we never see a 2-to-1 complex. So what we realized that the iron protein MoFi protein interactions, as well as the ATP hydrolysis process, are both asymmetric. So they are directional, right? The hydrolysis of ATP is not a concerted process. So then the important thing was, okay, now that we have turnover relevant iron protein MoFi protein complexes, are there any structural changes within MoFi protein that we were not able to see from static crystal structures before that may tell us something about the reaction mechanism? So we looked a little bit more closely at these turnover complexes. So complex one, I call pre-ATP hydrolysis, as it contains two ATP molecules. And complex two is mid-ATP hydrolysis, because it contains a single ATP and an ADP. And these color maps are based on B factor. So the redder something is, the more flexible or the less well-defined a region is. And the bluer they are, the more well-defined they are. And what we saw is that, of course, near the nucleotide binding regions, there's a lot of flexibility consistent with the conformational flexibility of the iron protein. Interestingly, we saw very few changes and very little structural conformational dynamics in the MoFi protein half that is bound to the iron protein. But we saw a lot of movement in the subunit that is about 100 angstroms away from the iron protein bound half. So what we saw in particular was this alpha-3 domain that forms a lid right on top of the catalytic FeMoco here that became very, very flexible that we had not seen before in crystal structures. So there was a lot of dynamics of this alpha-3 domain, perhaps implicating an opening and closing of this lid during the catalytic process. So that was a first insight that we had never seen before. And we also saw some distinct conformational or side chain conformational changes near the vicinity of these clusters. So one thing we saw was this triad of histidines that are right by FeMoco. So I'm showing you here two pictures. One is the proximal half of FeMoco that is bound to the iron protein. So this is the proximal half. And distal half is away from the iron protein that's bound. So in the proximal half, when we look at these triad of histidine residues that are near FeMoco and overlay it with the resting state structures, you can see that they are the same. They're the resting state conformations. But when we look at the distal side, we see that these histidines are flipped and different in a concerted fashion and found in a different conformation that actually puts one of the histidines into hydrogen bonding distance from one of the belt sulfurs in FeMoco, which could be a mechanism essentially to protonate the cluster, thereby increasing its potential so it can accept electrons. So that could be the gating mechanism. We don't know, of course. What we also saw was a cis peptide that is tryptophan 253. And the proximal side, this 253 is in the resting state conformation, but in the distal structure near FeMoco, this tryptophan assumes a very, very distant conformation that actually implicates changes in substrate access to FeMoco. So when we look at the resting state structure of this tryptophan 253, that actually is right near a substrate access tunnel that leads from the surface, shown in these purple spheres leading all the way to FeMoco. But in this alternate conformation of tryptophan 253, this substrate access channel is diverted to a different side of FeMoco. So that is, that also implicates perhaps iron protein dependent changes in the conformational state of the distal subunit to enable electron transfer activation as well as movements in the substrate access channel. But most importantly, we want to see what is happening at FeMoco during turnover. So when we look at just the distal, the proximal state where we did not see too many changes, and we map the electron or the Coulomb density in the resting state, the pre-ATP hydrolysis and mid-ATP hydrolysis states, you can see that the electron density or the Coulomb density, does not change. So you can see the homocitrate, nice electron density, the FeMoco, nice electron density in its histidines, and there's no change as we're proceeding through ATP hydrolysis. But when we look at the distal subunit, where a lot of action is going, you can see, again, the resting state structure in the free MoFe protein. Again, all the densities are very well defined. But suddenly, when we go to the pre-ATP hydrolysis and mid-ATP hydrolysis states, you can see that the Coulomb density near the homocitrate, which is bound to the molybdenum here, and the histidine, do not now necessarily go with or are appropriately fit to the resting state conformations of homocitrate and histidine. So that tells us that there is a lot of activity going on near this molybdenum end, homocitrate end of FeMoco that actually can implicate catalysis. So these blobby structures are essentially probably superpositions of many different intermediate states that are found in solution, and they do not necessarily fit with the resting state coordination of homocitrate and histidine. So that implicates also molybdenum in actually activation of nitrogen. So now, unfortunately, we don't see any nitrogen bound. We don't see any substrates or intermediates bound, and we certainly cannot see hydrogens bound at this resolution. But at least it tells us, these structures tell us that we can start to capture really this very enigmatic protein and its cluster at during turnover. And what we're hoping to do now is to see if we can find we can tweak the turnover conditions so we can get substrates and inhibitors populated in large enough abundances so we can see them with Cryo-EM. So basically,
this is Mike, and I have a question that might actually be relevant for this part of the talk. So that the question is, how do you account for difference in resolution when you're comparing the structures? That is, you have 1.8 and you got 2.3, you've got a mixture of several different resolutions. and saying, okay, this difference is difference in conformation and not just resolution.
Yeah, that's a very, very good point. We know that this is not a resolution effect, because when we look at the surrounding residues at this 2.3 angstrom resolution structure, you see that they're very, very well defined. Right. So we know that these changes that we see near FeMoco are not due to, loss of resolution. It is due to activity that is specifically happening, or conformational changes that are happening near FeMoco, because the rest of the protein is quite well defined.
Thank you. Good.
So again, this is just the beginning of, I think, our story. This is the first time that Cryo-EM had been applied, but more importantly, during the turnover state of nitrogenase. And we've already learned that asymmetry and directionality is pervasive in nitrogenase catalysis, whether it's ATP hydrolysis or the iron protein movement on the Mo-Phe protein surface and iron protein Mo-Phe protein interactions. But we also see, for example, that from Cryo-EM, that Mo-Phe protein is actually very highly dynamic, which is counter to what we saw before through crystallography, through static images. And we also believe that our structures show that ATP and iron protein the binding of that appears to induce conformational changes, or maybe or freeze conformational changes in the proximal subunit and free the distal subunit for conformational changes that may be involved in electron and proton transfer gating, substrate access, as well as FeMoco transformations. So that's the first part of my talk, is a success story that originated from work that we did at S2C2. And now I'm going to force a connection to another aspect of our research, and probably a little shorter second half. And that, again, we can use nitrogenase as a segue to the second half. So again, this is another view of nitrogenase, and it carries out a very complex multi-step reaction that involves a lot of conformational dynamics that are gated by ATP-dependent conformational changes. So this is a really, really complex machine. And of course, this very, very important machine that was crucial for the evolution of life on Earth, because it was the only source of fixed nitrogen, well, of course, didn't fall from the sky, right? I hope nobody in this audience believes that this thing was made as is. So it had to be made from smaller pieces. So if we do a retrosynthetic analysis of retroevolutionary analysis of nitrogenase, and we break it into its small parts, we realize that all of the subdomains in the nitrogenase complex, whether it's the iron protein or Mo-Phe protein, are Rossmann-like. So they have domain structures that are like P-loop or Rossmann-like secondary or tertiary structures. So it's almost as if the structure was made from similar Lego building blocks that were templated around these very, very funky iron sulfur clusters to do their reaction. So we can go even further back, and just maybe 4 billion years ago, even before Luca, saying that perhaps we had simple peptides like these beta alpha beta motifs that have very low functional diversity, as much complex chemistry as you can do with such small peptides, that eventually assembled into tertiary folds like the Rossmann fold, which are now bigger, and they can do more complex tasks such as recognition, binding, ATP nucleotide binding, electron transfer, and catalysis. And eventually, over another billion years or so, they evolved into nitrogenase, or when the same fold that nature evolution has used in different contexts also gave us diversification of functions to different types of supramolecular and quaternary assemblies, which now gives us potentially near infinite functional diversity and enables these types of complex architectures to carry out complex and multi-step reactions. And Of course, these are only some of the outcomes that you can get with a single building block. You can think of many, many different permutations that you can get. So based on this, we thought perhaps we can build a lot of complexity as represented by such extended assemblies using simple building blocks if we can do this in the laboratory ourselves rather than relying on evolution that takes billions of years. And so how do we take a building block like this and form arbitrary structures with desired functions or desired properties, in a way that a chemist would think about it? So evolutionarily, we can actually ask the question, how do we accelerate the generation and evolution of new and functional protein structures from simple building blocks? And the chemical answer to this is, and also the evolutionary answer, is to find efficient ways to form stable and selective interfaces so we can start assembling these things together. Now, if you take a look at a typical protein-protein interface in an assembly like microtubules, so this is one of the interfaces in microtubules, which is a cytoskeletal assembly, what you see are that these protein-protein interfaces in natural assemblies are very complex. They're heterogeneous. They cover a very large surface area. They have ordered water molecules, very, very complex set of hydrogen bonds and hydrophobic interactions that together give you the stability as well as the specificity, as well as the dynamics of these protein-protein interfaces. So such an interface is very hard to design from scratch. Although there are many, many advances in protein design, we cannot make a microtubule or something that is conformationally flexible, for example. So being inorganic chemists, by inorganic chemists, we thought that perhaps we can circumvent the complexity of making design such complex interfaces if we just link different protein building blocks together by either metal coordination or reversible covalent interactions. And because these types of interactions are a lot stronger than enthalpically stronger than these non-covalent interactions, then we would use only a very, very small footprint for design to enable these proteins to come together. And because we can control the details of metal coordination or covalent non-covalent interactions, we can also control the directionality and strength and symmetry of the complex structures that we built, as well as the dynamics of these protein assemblies. So, as a proof of principle, we used this 4-helix bundle protein, a monomeric 4-helix bundle protein, as a building block. So just like a Rossmann fold, 4-helix bundles are also very ubiquitous. And this protein that we use is a cytochrome, so it is a heme group, and it is a monomeric protein, and we want to see if we can build larger architectures from it. So using metal coordination, for example. And of course, if I highlight all of the amino acid residues on the surface of this simple protein, what you quickly realize is that essentially any hydrophilic residue on the surface of a protein can coordinate metal ions, and very, very strongly so. So how do we control discrete self-assembly of such an architecture into the structures that we want? So what we then did was to place histidine residues that are 1 helix turn apart to give us these bidentate chelation modes that we thought would nucleate metal ions more selectively than if we just had monodentate ligands here. And we made this building block with 2 pairs of histidines. And what we saw was that when we added nickel 2 to this, we got a C3 symmetric trimeric assembly. So 1, 2, 3, this is a crystal structure, and that's dictated by the nickel coordination. It turns out nickel 2 likes octahedral geometry. So it likes 3 pairs of histidines around it to get a trimeric structure. When we want to call a copper 2 is next to nickel in the periodic table, but it prefers a square planar geometry. So what we saw was that copper 2 imposes C2 symmetry to form a dimer now that has 2 pairs of histidines in a square planar geometry. And we want 1 to the right in the periodic table, that's zinc 2. Now zinc 2 has a D10 electronic configuration. It likes a tetrahedral geometry. And what we found was that it imposes now the formation of a tetramer with that has D2 symmetry, because all of the zincs like to find a tetrahedral combination of amino acids. So this is a very, very simple inorganic experiment in that we take a protein building block, or the ligand that we call in organic chemistry, and we can change the geometry of the complex that we form by changing the metal ions. But for a biochemist or protein designer, this is a significant step, because there's no other type of bonding strategy besides metal bonding that from a single building block, you can get very discrete multiple outcomes just by changing something in the solution. So this took only four mutations on the surface. So we thought, hey, we can actually use metal coordination to build tailored architectures. And we can actually build a lot of functions starting from these types of architectures. We can make enzymes, we can make allosteric assemblies, we can make new types of metal coordination sites. But we also saw was that when we place and design the surface of this protein for metal binding in a different way, we can also get 1, 2, and 3-dimensional assemblies. So here is actually the first Cryo-EM structure that we solved that is from 11 years ago. This was way before Cryos, et cetera. But this got us into Cryo-EM, but also showed us that we could make these microtubule-like or S-layer-like dimensional assemblies that are not discrete. So these types of materials. So then we thought, hey, can we use these strategies to make any arbitrary extended architecture material with distinct dimensionalities? So can we make cages, for example, that have point group symmetries, or 2-dimensional crystals with plane group symmetries, or 3-dimensional frameworks with space group symmetries that we can actually do crystal diffraction, X-ray diffraction on? And the key to making such architectures is to combine different symmetry elements to get periodicity over certain dimensions. So for example, to get a tetrahedral architecture that is a point group symmetry, you can combine C3 symmetry with C2 symmetry in a certain angular relationship, 54.7 in this regard. You can take a C4 symmetric assembly and combine it with C2 symmetry to get a planar assembly, or you can have an octahedrally symmetric building block and you can combine with C2 symmetric connections to get a 3-dimensional lattice, a cubic lattice. So what we wanted to do is use these types of strategies to make these types of architectures. So the story I'm going to tell you is about making a cage using metal coordination. And the reason why we're interested in making cages is because, first, they're aesthetically very, very pleasant, but also they have functions that stem directly from their structure. So, for example, this tetrahedral architecture, or this octahedral architecture, or this icosahedral architecture, they have all hollow interiors that enable them to carry out site selective reactions or protect their cargo from the outside. So their form really fits their function, and they're aesthetically very, very pleasant. The thing is, these multimeric assemblies are composed of a single type of building block, but that simple building block is monomeric, so it has no inherent symmetry. So it has to form many, many symmetric interactions with their partners to give you these types of point group symmetries. So, for example, if we take ferritin, which is an iron storage protein that has octahedral symmetry, so 432, it's made out of a 4-helix bundle protein. This is the surface representation, and 24 of them. And each monomer has to have a surface patch that forms its C2 symmetry. It has to have 2 patches, shown in blue here, to form a C3 symmetry. It has to have 2 other patches to form C4 symmetry that, like puzzle pieces, all come together to form this 432 symmetric octahedral unit with a hollow interior. So the problem is, how do we take an asymmetric monomer and design multiple associative surfaces at the same time so that it assembles into the architecture that we want? So we decided to use metal coordination for this purpose again. And again, going back to what I showed you before, as I explained, we can take a single building block with histidines on its surface, and depending on the metal that we add and the electronic configuration preferences of these metals, you can impose different symmetries in C3, C2, and D2. So for example, then you can say, hey, why don't you take the same building block and add nickel 2 and copper 2 at the same time to see if you can form C3 and C2 symmetry at the same time so that you can get, for example, a tetrahedral architecture. The problem is one of selectivity, because both nickel and copper and the histidines are what we call soft or intermediate soft metal ligand combinations. So that means there's no selectivity. If I add nickel 2 and copper 2 to this building block at the same time, I would just get a mess, because these histidines have no preference for nickel over copper 2. So there's no selectivity that proteins actually get through these non-covalent interfaces. So how do we get selective metal binding and impose different symmetries at the same time? So what we want to use is the hard-soft acid-base theory, which says that hard ligands such as carboxylates like to bind hard acids like 3+ charged metal ions, and vice versa. Soft ones, nickel 2, copper 2, like to bind things like histidine. So how do we find a hard acid base combination? And we again took inspiration from nature. It turns out bacteria, for example, to grab iron from the environment or from our guts, they use these agents, natural products that are called siderophores. So siderophore comes from iron carrier in Greek. And iron 3 is hard, so it has 3+ charge, and it likes to bind these functionalities that are like catechol, they're oxygen rich, or hydroxamic acids. So these bind iron 3 with very, very high affinities and with very high selectivity over soft metals like iron 2 or nickel 2 and copper 2. And importantly, they also impose C3 symmetry, as you can see. So these are all octahedral structures that have coordination that has C3 symmetry. So we decided that if we could actually take our protein building block and, for example, design a zinc binding site to impose C2 symmetry and combine it with, for example, a hydroxamic acid site that can bind iron 3 selectively in a different position that could impose C3 symmetry. And we combine these symmetries, we can actually get a tetrahedral structure. So to do that, we first synthesized a non-natural amino acid. So we synthesized iota hydroxamic acid that selectively goes after thiol groups of cysteines. So we can make this hydroxamic acid derivative that we call cis-hydroxamic acid that is as long as an arginine, essentially. And we took the same building block, 4-helix building block, and designed a zinc binding site on one surface and hydroxamic acid attachments on 2 other surfaces to give us C3 symmetries. And we first checked what they form in solution. So this is an analytical ultracentrifugation plot of different species. So the monomer is the monomeric protein is at 12,000 atomic mass units. And when we add only iron 3 to this, we get essentially what amounts to hexamer. And when we add only zinc 2 to it, we get a broad distribution around the 9mer. But when we have iron 3 and zinc 2 at the same time, in that case, we get a very, very, nice narrow distribution that corresponds to a dodecamer as we want. And when we looked at the solution structure that is formed, what we saw was a very, very high population of these donut-shaped architectures that you can see in very, very high, low polydispersity. And of course, donut shapes are characteristic of cage-like architectures. So then we turned to Cryo-EM And the cryo-EM of these architectures was not nearly as complex as that of nitrogenase under turnover conditions, because these are very uniform structures. There's only a single species, and they are also very highly symmetric. So we did some simple single particle reconstruction, and with some tetrahedral symmetrization, we were able to get a structure that's around 2.6 angstroms. And this is what the Coulomb density of the architecture looks like. You can see all the helices and amino acids. Now, of course, we could also crystallize it, the structure. And when we compared the crystal structure that's formed with the cryo-EM structure that is representative of the solution structure, we saw that the cryo-EM structure of the tetrahedron was a little more expanded. So perhaps the lattice compresses this cage, whereas in the solution state, it's a little bit more flexible and it can open up. But long story short, this is the structure of the assembly that is formed. It is composed of 12 proteins and 8 iron 3s that specifically bind in these C3 symmetric corners, and 18 zincs that actually make up the C2 symmetric interfaces. So to me, as an inorganic chemist or supramolecular chemist, this is pretty amazing, because if you think about it, this is 3 different species and 40 components that with very high fidelity self-assemble in solution to give you this very, very tightly packed cage-like, almost virus-like architecture. And that is also stimuli responsive. So there's a lot of interest, for example, using a Rosetta, others, to make cage-like architectures that could be used as, for example, antigen displays or storage of other proteins. And the nice thing about this this cage-like assembly is that it's stimuli responsive, because we can control the assembly and disassembly through metal coordination. So this is so tightly packed that we actually found that we can even store small molecules on the inside. So we can put rhodamine in, and after 4 days, the rhodamine fluorescence is still contained within this architecture. So there's very, very few gaps, so we can really store it. And after 7 days, about half of it remains. But importantly, when we add a chelator or a reductant to release the iron 3 or zinc 2 ions, immediately the container or the cargo is released. So this gives us a good way to make these types of very tightly packed cages, virus-like cages, that we can hopefully use in, for example, drug storage and release. So that is the second story that I wanted to tell you. But of course, there's other structures, other types of projects that we work on in the group that we also use cryo-EM extensively for. Here's an example of, just as a glimpse, we want to see if we can also use, for example, DNA Watson-Crick-Franklin base-pairing interactions to further control self-assembly. So this is the same building block attached to a single-chain DNA with its complementary group, and these actually form 2-dimensional crystals. And 2-dimensional crystals you can not do crystallography on, but we can do cryo-electron diffraction on. So we could get a 6 angstrom structure that showed us from the reconstruction, the symmetry and the arrangement, these volume maps that we could use to actually determine the molecular arrangement in these 2-dimensional structures, where you can see each of the proteins that are attached to one another through Watson-Crick base pairing, and they're further connected to one another through metal coordination interactions. We can also make for example, new dissipative ATP-dependent nanotubular assemblies like microtubules. This is an example where unpublished example, where we take a AAA plus protein and convert it into a nanotubular architecture that requires ATP hydrolysis, just like microtubular actin, to form these nanotubular architectures whose structure that we determined through cryo-EM helical reconstruction, about 2.5 angstrom resolution. We also work on artificial ways of controlling nanotubular assemblies, such as light responsive. So this is an assembly, nanotubular assembly, that is formed through azobenzene, cyclodextrin, supramolecular interactions that can be made or broken by UV or visible light. again, about a 6 angstrom cryo-EM helical reconstruction that tells us what the arrangement of these architectures are in these nanotubes. Again, all from data we've collected at S2C2. Anyways, just the summary of the second part of my talk is that I hope I showed you that we can use basic tools of inorganic and supramolecular chemistry to make complex biological assemblies, and that we can use strong and irreversible bonds, such as metal coordination, disulfide bonding, to create dynamic structures that are also responsive and reconfigurable. But most importantly, the ever improving cryo-EM and electron diffraction methodologies are really making this line of research possible, because we would never be able to solve those types of structures and address the types of mechanistic questions that we do with nitrogenase, just relying on other spectroscopic or crystallographic methods. And I'd like to thank my group. I guess I'm running short on time, but importantly, I'd like to thank Hannah, who was a graduate student who worked on the nitrogenase cryo-EM project, along with Mark Herzig and Brian Cook in his lab. And Tim Baker was our was the first group, a person who taught us how to do cryo electron microscopy. And for helical reconstructions, we got a lot of help and guidance from Ed Eichelman's group at Virginia. And of course, I'd like to thank SSRL and S2C2 for extensive crystal data collection, as well as SACS, but also for cryo-EM. We're using essentially SLAC to its limits. We're using every tool that they're able to give us, and we're really grateful for all their resources and their helps, and all the Beamline scientists as well. So thank you for your attention.
Well, thank you, Akif. That was amazing. That whole story is amazing, and thanks for the shout out to S2C2 and to SSRL and the other facilities that we hope will make S2C2 and SLAC a one-stop shop for structural biology. Maybe you can get on LCLS and do things at nanosecond timescales with the free electron laser.
Do we have time to answer questions?
Yes, there are a couple of questions in the Q&A, and if you want to put questions in, just stick them in the Q&A, or that would help. One question was, why not use cysteine as your hook to coordinate metal?
We actually do use cysteine to hook metals, but not in this context, but in the context of what I talked about. Mm-hmm. We do use cysteine, but of course cysteines also tend to form disulfide bonds, so that's another complication. But we have actually used cysteines extensively in the disulfide bonds to actually make extended structures, protein structures, instead of metal coordination itself. And the beautiful thing about cysteine disulfide bonds is that it's also a reversible covalent bond. So we can control that equilibrium through the redox potential of the solution. And we've made a lot of assemblies, but I didn't have time to talk about those, and none of them really fit under the cryo-EM umbrella. So that's why I did not talk about cysteine. Okay. I don't see. I have a question too, or a question or two. In all of your work, and it seems like there are one recurring theme is that an interface, certainly for the iron sulfur interaction with the nitrogenase, the interface should not be too strong. That it shouldn't be kind of so strong that it's hard to dissociate or change. And so do you see a characteristic kind of weak and greasy interaction that's different from really strong interactions like we would hope for, like antigen antibody complexes that really say, okay, this forms nicely, but it can also fall apart easily too? I mean, that is really the essence of what we try to do with, that gets to the essence of what we try to do with metal coordination, is that, I mean, you point something out that's very, very important. You want those bonds that you form between proteins to also be very labile. So labile is the opposite of inert, in the sense that it's a kinetic term that, when things come together, they should also be able to come off very, very quickly. So, and that's what metal coordination accomplishes. That's what you were asking, right? So,
well, I was actually, in terms of the protein-protein interactions that also have to be kind of slippery and form and break. And it looks like it kind of looks like in the iron sulfur case that you have maybe one of them binding to one end, and then it switches and binds the other end, and they shuttle back and forth, and that implies, and it's great because Cryo-EM can do that in silico, conformational flexibility. But yeah, it really says that these interactions are very labile and very transient, but good enough.
yeah, I mean, they are transient. I guess they're long enough lived, with sufficient stability that we can capture them, as they form, yeah, I mean, there may also be very, other types of intermediates and conformations that are not strong, that are populated in a solution that may perhaps not withstand, freezing and vitrification. We do not know. So it's all a beginning. For all the things that we were able to capture, we're probably not able to capture 1,000 other things. So, we're in the beginning. I mean, obviously, we're going to need a lot more technology development, technique development to be able to, get all the details, for sure.
Yeah, one more question from the Q&A. Did you try to get the Cryo-EM structure for the whole complex in the absence of substrate N2?
Yes, and we're also working on getting them, in the presence of inhibitors, et cetera. And yeah, I mean, the sad thing is we have not been able to yet see any N2 bound, even when we have N2 in solution. But I think that's a very, very important question as to, like, if there is no N2 in solution or in the environment, are we going to get different types of architectures that may, for example, what happens when the system is driven for only hydrogen production rather than N2 fixation? We may get different architectures. But yeah, the problem with this system is that even the data transfer takes a long time, because there's so many images and data processing, and really, it was a heroic effort by Mark and Hannah and Brian to be able to even get 3 distinct structures out of that, right, really heterogeneous mix. So it's a huge data processing and data analysis problem that we have to get better at. Yeah, but we're trying different conditions, different substrates, different inhibitors, different amounts of reductants, so we can capture and populate different intermediate states and in different abundances. So we can maybe piece together all the intermediates and the catalytic mechanism.
Great. Any other questions? I don't see any other questions in the Q&A. We maybe go back to Ed to wrap it up.
Okay, well, thank you for attending, and we'll see you next time.
All right, thanks
Thank
again.
you.